Hello and welcome back. We have a two-part series on the nervous system. We're going to be starting with the central nervous system and then moving on to the peripheral nervous system. But then that's it, guys. You did it. You've done it. We're here. We're at the very end. You should be so proud of yourself. Thanks for sticking with it. This is not an easy subject. I'm so proud of all of you guys. And without further ado, like I said, let's just get started, and you guys are almost done. So congratulations, and don't forget to study for your final. Check that you've done all the missing assignments, that you've turned in anything on Canvas, including extra credit, and please email me if you guys have any questions. All right, let's get started. Now, the function of your nervous system essentially is to monitor outside responses. So certain stimuli that you're going to get from the outside environment, your body's going to interpret these signals and then decide what is a proper response to that. And that's really all our nervous system is, via our central or peripheral. Now, really what we're doing is these sensory receptors are monitoring the changes inside and out of our body. And then these processes are going to be basically interpreted by our brains. And then eventually it's going to dictate a response by activating certain effector organs to respond to whatever our outside stimuli was. So essentially that's what we see. We see water. That impulse is then going to get sent to our brain. We're then going to interpret that message. We're going to send our signal or dictate a response, which is going to be grab that glass and drink said water, right? We are thirsty. We see the water. We're going to reach out with our hand. We're going to grab that water and we're going to drink it. So this is again, stimuli, which properly interpreted is going to exhibit a response, and that's what we can see. So this input is known as sensory input. The integration is also that, again, you're trying to figure out what to do, right? That interpretation. And then your motor output is going to be that dictated response. What are we actually physically accomplishing? So the two basic divisions, the ones I already talked about, are going to be the central nervous system, or the CNS. This is going to include your brain and your spinal cord. And then you also have your peripheral nervous system, which is going to include things like your nerves and your ganglia, which essentially is just a big cluster of these neuronal cell bodies, which we're going to go into more in just a second. Now, we do have both sensory and motor signals. So essentially your sensory signals are your afferent, right, coming in, being interpreted by the brain, and the signal is going out. That's your efferent, that's your motor signals. So again, sensory is input, right, coming in, the signal's coming in, and then efferent is going to be your motor, your response, your dictated response, whatever that is. So signals picked up by the sensory receptors, and they're going to be carried by the nerve fibers to the central nervous system from the peripheral nervous system. So again, those nerves, those ganglia, they're going to be the one picking up the response, and it's going to be sending it to the central nervous system to decide, okay, now what do we do? Great. And let's exhibit our motor signal, which is going to be carried away from the central nervous system, and that's going to innervate certain muscles or glands, whatever we're, again, trying to accomplish, whatever goal we're trying to accomplish. All right, so we've got our sensory nervous system, our afferent. We've got our motor nervous system, our efferent system. So coming in, we have sensory receptors. Obviously, this is going to be receiving our signals, whatever signals we're receiving. This is going to transmit the information from those receptors to the central nervous system, right? That central nervous system is then going to dictate a response. Now, these signals are going to be either somatic sensory signals, meaning coming from our skin, our joints, our fascia, our skeletal muscles, or our special senses. Or it's going to be our visceral anatomy, or our visceral sensory, I should say, which is going to be sensory information from our viscera, from basically inside. Now, our motor nervous system, again, is going to be transmitting the information from that central nervous system once we've decided what we need to do, once we've interpreted. We're going to have integration, right? And then, therefore, we're going to dictate our response after that's our motor nervous system. So this is going to send the motor information to the effectors, who's actually doing the reactions. So we can also have the somatic motor, right? This is going to be your voluntary nervous system. It's going to innervate mostly skeletal muscles. And that's what really, okay, this is my dictated response. I need to move. Or it's going to be your autonomic motor system, which is basically involuntary nervous system. Things like your cardiac muscles, your smooth muscles, your glands, everything internal, basically what needs to happen if we're, say, low on something, or we need to lubricate something, hence, secretory glands. Whatever we need to do, again, is going to be dictated internally by that autonomic. And that just basically means we're not controlling it. We don't have to think about it and be like, glands, go, right? Well, that's not how that works. It's basically just happening on its own. All right, so let's zoom in and see what we're actually looking at here. And that is our neuron cell, very clearly defined right here. You can see the nucleus in the center, and you can also see some of these little support cells. These are known as neuroglia cells. And essentially they're just glial cells, specific types, which we are gonna learn about and you will need to know. But these basically are just gonna be supportive cells. That's essentially what they are. And then your neurons are the ones that are actually transmitting those electrical signals. So those impulses that we need to, to send out those signals, that's all coming from your neurons. Let's see. Now, these support cells or these neuroglia are not excitable. And so essentially what this means is we can't trigger them with these electrical impulses. So these neurons can be excited. Remember, that's like your neurons like firing, right? But these guys are not. Essentially what they're doing is they're just surrounding and wrapping the neurons. And there's going to be a lot of things that we see that are basically insulating the neurons because you have this electrical impulse. You don't want to lose it. And you don't want that signal to be lost anywhere. You do want it to go as far and as fast as it needs to go. All right, speaking of our good friend, the neuron, which we've already seen once before, but we're going to get into in greater detail. This is our basic unit of cells for the nervous system. And these are going to be specialized cells that conduct those electrical impulses that I talked about. That's our signaling. We're basically just one big walking source of electricity that's firing on dee, 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 almost like Morse code, sending off these signals through our nerves. And these nerve impulses are known as action potential because it is the potential to make action happen. And that's really what motion is, or response is we're making some kind of action happen and therefore action potential. So as these get signaled, right, that electrical impulse gets signaled, the signal is going to be received here and then it's going to fire again and send it all the way down here through these little, these little myelinated sheaths right here. And that's going to help move those signals along. Now let's see. I think that's pretty much it? No. Okay. Now, the neurons themselves pretty much last our whole life. Like from the time we were developed, this is going to live and they're going to function the whole time, which is really rare of cells. Most of our cells die off after a while and they get replenished via mitosis or meiosis, right? But not these guys, which is why we don't want to damage our brains because we only have one set of brain cells. And so this is why head trauma can be so disastrous, is because essentially if you lose the function of these neurons, they don't really regenerate. They can't really come back. Because again, they don't divide, they don't go through these cellular processes like mitosis, right? So basically what we have is, we're able to do them when we're growing and developing, when we're still a fetus, but once we reach a certain point of maturity, then that's it. We've got the ones that we've got. Now, neural stem cells are an exception. There, you might have heard of stem cell research and all this kind of stuff. We are testing a lot of these different things out because these regenerative cells that are known as pluripotent, or that can become many different types of cells, it's really, they're really great and there's so much potential there, but we're still just diving into that. Now, because these neurons live for so long and they're really, they're firing all the time and they're firing as efficiently as possible. Again, they are controlling all of our processes and keeping us alive. So important that they do so. But they do have a very high metabolic rate because of that. So essentially they are going through a lot of food. They're going through a lot of oxygen. They're basically doing cellular respiration really, really quickly. So they're also creating a lot of metabolic waste. And therefore, we do have cells that are going to come and support them as well that are basically just cleaning up after them. They're like, here, what do you need? I have food. Cool, let me pick up your trash. Just so that they can, again, keep constantly firing as efficiently as possible. Now, unfortunately, because they have such a high metabolic rate and they do need so much oxygen, when depleted of oxygen, the neurons will die after only 5 minutes. This is why if you are, say, drowning or you're suffocating or you're being choked or whatever, you basically lose your oxygen, you have about 5 minutes before it's too late and you start to have brain damage because you start to have these neurons die off. And that's really all it is they're waiting for oxygen, and because they need so much of it so quickly, when they don't get it, they die pretty quickly. So this is why you have about 5 minutes. It's very important if you are someone who's drowning or something, get them out of the water, get them CPR, right? Do the pumping and the staying alive, right? It's because you need to get the oxygen to these neurons. And that's why they say your brain needs oxygen. Essentially, it's these neurons that actually really need the oxygen. Now, let's go into the parts of this neuron. So we talked about how this is the nucleus right here. Inside this big part, right? That big part is the cell body. So it's the cell body of the axon. It's really like the big one. Now this right here is your axon right here. This guy, whole thing would be considered the axon, right? Right, this is your axon hillcock as it gets smaller and smaller and smaller here. Right, these guys are dendrites. These are essentially the receptive regions, which is why there's so many little branches here. They're reaching out and they're going, hey, I'm waiting for my signal from whoever. And then once they get a little signal triggered, it's going to send the signal in and then down through the axon hillcock, and then all the way through these right here. Each one of these little sections are known as Schwann cells. Right, these little Schwann cells are wrapped in what's known as a myelin sheath. A myelin sheath is basically going to be an insulation. It's kind of like wrapping a wire with that little rubber. So one, you don't touch it and get shocked. And two, so it keeps everything insulated so that your electricity is passing through uninhibited. Now, each of the gaps right here in those little myelin sheaths, because they are like little separate pockets, right? These are known as the myelin sheath gaps or the nodes of Ranvier. Let's see, what else do we have? We have our, these are our terminal boutons. So you can kind of think of this as like the incoming region, and then the signal goes all the way down here, always going this way to those terminal boutons. And that's the secretory region. So essentially it's going to be the one secreting the signal to the next cell, which is then going to be received here and then transported to the next one as needed. Let's see, we also have some neurofibril right here. This is that little connective tissue. What else we've got? I think that's pretty much it for that. So cell body, right? Nucleus, dendrites. This is considered the axon hillcock coming down into the axon, right? This is the Schwann cells here, right? Myelin sheath gaps. Boom, 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 because the Schwann cells are basically just wrapped in myelin. And we're going to learn how that happens in just a second. And then finally, you have the terminal boutons out here. All right, so starting with the cell body, looking a little bit more in detail. This is also known as our soma, which again is going to be this section right here containing that nucleus. This is really the cell's control center, which is why it's the cell body. It's like the bulk of the cell and the most important parts. Let's see, it does stain darker when you're looking at it because of the ribosomes. Again, ribosomes are going to be creating proteins, so very important for these guys. Also creates gray matter in the brain and spinal cord when you're actually doing analysis of that. What else? Again, directions are going to go in here, right? Impulses come in, traveling down here and going out here. Again, moving down always in this direction towards those terminal boutons. Now, most of the neuronal cell bodies are going to be located in the central nervous system. This makes sense because this is your brain, right? This is where all these signals are coming from, and then boom, you're going to be interpreting and sending them back out. There are some neuronal cell bodies in the peripheral nervous system, but those are known as ganglia. Those are ganglia, so those are a little bit different, where these are neurons. All right, so the dendrites, again, the impulse right here, the incoming impulse. Right, so that's what these guys are going to receive right here. So these longer ones are known as the dendrites. Off each dendrite are going to be dendritic spikes. This is essentially just increasing those areas where impulses can come in. So increasing the amount of signals that you can take in. And that's what you can see right here, as that impulse is going to, the direction of the impulse is going to go down here and then coming out through here. Now, as we move down to the axon, right, down through the axon right here is the longer part of the neuron. So the cell body and the axon coming all the way down through here. There's almost always one axon per neuron, but sometimes we do have multiple axons per neuron. And sometimes, like I said, we do have these little offshoots right here. The axon hillcock is basically going to be this connective piece from the cell body, right, to the axon. So that's that hillcock. And then this is going to be that axon collateral that I talked about, that little offshoot, right? That's anytime you have a secondary axon coming off, that's going to be that axon collateral, which essentially is just a branch of the axon. As you move down to the axon terminals, that's going to be this guy right here, right? And that's going to have our terminal boutons at the very, very end. Now, this is where we basically have these little synaptic knobs, and that's what you're going to see right down here. That's basically these little bulbous areas that are used to kind of secrete whatever signal that we're sending from one cell to the other. And this is really all just done through the cell membrane. And so it's just like what we've learned before, those little vesicles. It's going to be little transportation vesicles that are sending the signal where they need to go. And that's these synaptic knobs, these little knobs, those little nubbins right down at the bottom. All right, talking about the Schwann cells, or Schwann cells, I've heard them both, also known as neurolemnocytes, and that's these guys right here, right? And they are covered in these little myelin sheets. And this myelin sheets, again, is going to be used to insulate these cells. So it's also going to speed up the transmission because we're not losing any of that electrical signal. It's not kind of going this way or that way or dissipating outwards. It's basically being shot in a single line. And that's exactly what they do. And in fact, it actually jumps from one to the other, to the other, to the other. So because of these gaps, because of these nodes of Ranvier, because of these little gaps, essentially, it allows them to like speed up the transmission instead of dragging all the way through. It's almost like teleporting from one to the other, to the other, to the other. It's actually pretty cool how it works. Now, these myelin sheets, again, are just going to be these neurolemnocytes, which are together on one neuron, and that's basically just going to be folding in on itself. And we do learn how this happens in just a second. Okay, so we've got our neurofibril node, which is this section right here, also known as the nodes of Ranvier. We've got the neurolemnocytes, which also is the Schwann cells. Sorry that there's two names for everything, but you need to know it. I'm sorry. And then finally, the myelin sheaths, again, is just going to be this multi-layered wrapping of the membrane. All right. Now, I did mention that these cells are going to be connecting from one to the other, and that's essentially how they talk to each other. And so what's going to happen is it's known as synapsis, and that's what we're doing is we're creating a connection between the two. So it's the site at which these two neurons are going to communicate is the synapsis part. Now, most of the messages are going to be sent via chemicals. Again, we're not going blah, 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 blah. That's not how we communicate, or at least not how cells are communicating. So we actually do this through chemical signal. So that's what I mean in those transportation vesicles. We're actually going to be secreting that signal via whatever chemical we need to release, and that's going to trigger our proper response. So we do have our presynaptic neuron. That's going to be anything before, right, that signal is being released. So it's going to be conducting the signal towards the synapse, the connection point. And then our postsynaptic neuron, which would be the second neuron that's receiving the message, and that's going to be after the connection site. So it's kind of like two neurons here. This would be the presynaptic, this would be the post, because the signal is going to jump, right? And that synapsis, again, would be that little connection point. All right, so that's what we can see right here. So this is going to be one terminal bouton, right, from this axon connecting to this dendrite of this neuron, and that's going to be the synapse part, right, the connection part. And then the signal is going to come down through here, in through here, right through here, and then out through here. And this would probably connect to another dendrite of another neuron. Now, we do have two different types of synapses. We do have the axodendritic, which is between the axon terminal of one neuron and the dendrite of another. That's the one I just showed you. That is the most common type of synapse. But we also have an axosomic connection, and that's between an axon and the neuronal cell body itself. So not at those little dendrites, but actually the cell body is where it attaches. Now, how it actually is going to send one signal from the presynaptic axon to the postsynaptic axon is just this. So this is considered to be our presynaptic axon as we're ending here, right? This is our synaptic cleft meaning kind of section like a cleft palate, right? Those little sections, or cleft chin, like the little butt chins, right? So this is your cleft right here of your synapse. And this is going to be your postsynaptic dendrite here, the one receiving the signal. So the nerve impulse is coming down through here, right? It's going to pass by a lot of mitochondria because remember, we need all of the sugar and we need all of the oxygen. And who's going to do that for us? Good old mitochondria, right? It's going to release whatever chemical signal via these synaptic vessels right here. And that's what you can see right here. These are the vesicles that are releasing the neurotransmitter. That neurotransmitter, again, is going to come down to the postsynaptic dendrite here. And there are usually going to be channels right here, and that's what's going to be receiving this. Or it's going to, again, bud just like this, create its own little vesicle and go in, just like this. Again, this is all membrane. It's membrane to membrane. We've done this before. Exocytosis, endocytosis is essentially what we're doing. We're going from one cell, we're releasing, and then we're going to be grabbing this and bringing it into the cell over here. All right, so let's talk about the different types of neurons. Remember how I said they always have one axon, but sometimes they have more. You've got your dendrites right here. You've got your peripheral process, leading to your cell body, and then you've got your central process, right, leading to these little terminal boutons over here, the synapse. This whole thing is considered the axon. So there's a unipolar neuron because it is going in a single direction with a single axon. There is a short single process right here that goes up to the cell body, right? But again, this is just one process connecting this axon to that cell body. And this is the most common one that we see in our peripheral nervous system. Now, if you have a bipolar neuron, right, bi meaning two, you have two processes from the cell body. So in this case, you've got your dendrite over here, right? You've got your cell body here, right? But you don't have it separated with that other cell process like this right here, like the short single process, right? You have your cell body right in the center. Okay, so it's going through this guy right here instead of having the little offshoot. It's just going straight through. And then you have your axon, which technically is only this part right here, whereas this is still considered one big dendrite. Now, this is very common you see in the olfactory system and the retina of the eye. And it's because these guys are actually working pretty hard and pretty constantly, especially the eye. We just learned in special senses how exhausting our eyes can get because they're constantly doing things in every second of every day, including when we sleep. But that's why they're found in the nose and in the eyes. Now, you can also have a multipolar, which basically you have many dendrites and one single axon. So in this case, we have dendrite, 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 dendrite. You have your cell body right here in the center, and then you have your axon just below that. And you can see right here. Or you have all sorts of this guy. Look at this guy. It looks like he's wearing a wig that's just freaked out, right? That's all of these dendrites are basically pulling these signals in and then sending them out just through this one single axon right off the cell body. So these messages are all coming in from all these different directions. And this is going to be by far the most common in our bodies because of our muscle system, because they're basically, this is how our muscles get signals. And therefore, this is how we basically control everything with our motor neurons. So this is going to be our muscles, our glands, anything like that, which is why it's the most common here in our whole body. All right, so sensory neurons, again, are going to be transporting impulses to the central nervous system from our peripheral nervous system, right? We've sensed it, it's come, we felt it, we're sending our signal to the brain, to our central nervous system. Basically, these guys are all going to be unipolar because we are sending them in one single direction. Go, go, go. This is, again, we need to interpret our signal and then we need to develop a proper response. Now, we do have cell bodies in the ganglia outside the central nervous system, which are basically these single short processes like that first example that I showed you guys right now, the little bulb guy coming off with the dendrite and the axon separate. Now, these guys do have a central process, again, that runs centrally to the central nervous system because it's super important. We need to get there. And we also have a peripheral process that extends peripheral to the receptors. This is where we're receiving the impulses from. So we do, again, have it coming in from the sides and then shooting straight to the central nervous system where we actually need to interpret. Now, the motor or the efferent neurons, again, are carrying those signals away from the central nervous system to our peripheral nervous system to basically, to react, right? Well, sorry, not to our peripheral nervous system, but to our effector organs, to the rest of our body, to our periphery, right? To everywhere else in our body to actually exhibit our proper response. Now, most of these guys are going to be multipolar because, again, we're sending signals in from lots of different places and we need to send them out pretty quickly as well. Let's see, we do form these junctions with these different effector cells because this is who's receiving the signal. And this is who's going to take it to whatever organ we need to produce whatever with. Now, we also have what's known as interneurons, which are association neurons. And they are mostly multipolar, but they're also confined specifically to the central nervous system. So these are just kind of like, not really supporting, but they're just kind of in between. They're kind of like a transition between the sensory and the motor neurons. Let's talk glial cells, because remember I did tell you you need to know them and you need to know their functions and to be able to differentiate what they look like. So let's get into that right now. Now, these glial cells are going to be very common. They're going to be found all throughout our central nervous system and our peripheral nervous system. These are also known as neuroglial cells. They are smaller than neurons. They are capable of mitosis. And again, they're really kind of helping the neurons out. So they're like the backup team. They're like the B team going, hey, what do you need? We're here to get it for you. They are typically distinguished by things like size, intercellular organization, presence of specific cytoplasmic processes, But we are going to go into just the ones we're going to talk about today, including the first one, the astrocyte, which is the most common. Okay, this is the most abundant of all the glial cells. And this is a little kind of like a star. Like imagine his center and then boom, he's kind of radiating out offwards. Always kind of reminded me of like a sea star, right? That's an astrocyte. Astro means star, right? So 90% of all the nervous tissue are going to be found in some areas of the brain. More than 90%. Like I said, these guys are really, really abundant. And essentially what they're doing is they are connecting the neurons to the capillaries. They're touching the capillaries and they're touching the neurons. So the signal needs to go from one to the other. We've got it. And these guys are helping. So especially for diffusion, right? Increasing that surface area can increase the amount of diffusion that we have, right? Those little vesicles that are going to be transporting all of that good stuff. Now we do have lots of surface area again, lots of projections, right? You can see right here covering this guy in the capillary, covering this guy, this neuron right here. These are known as perivascular feet. Essentially, they're just kind of like grabbing a bunch of different things and holding on. So they are touching the capillary walls and the neurons always right here and right here. Now, some of the functions, these guys are definitely multifunction because they're doing so many different things. So they're not only helping to replace damaged neurons. Remember, we can't really get them fired up again, but if there's anything damaged, we do want that removed. We don't want it anywhere interfering because if the signal's not being properly sent, we want to make sure that we get it out of there so that the ones that are working can fire properly. Let's see, we help with synaptic transmission, like I talked about, those signals that need to be sent, right? These are all being sent via these guys, well, via help with these guys. They regulate chemical composition within fluid within the brain. They help to form the blood-brain barrier, which basically prevents any unwanted material from getting into the brain. If we don't want any unwanted material getting into our system, we certainly don't want it getting into our brains. Let's see, we also help with development when we're a fetus. So these guys are also helping in the actual development of the original neurons that we get while we're developing before that all stops. And they really do help to strengthen and organize nervous tissue in the CNS. They're basically keeping everything running properly. Very important. Next up, we have the ependymal cells or epidemial cells. These are cuboidal epithelial cells that are found in the central nervous system. You can see the little cubes right here with all those little cilia, right? So those little ciliated right here. They do line the central canals of the spinal cord and our brain vesicles. Very important. Together they do form this plexus. We've seen lots of plexus before, but this one is known as the choroid plexus. And essentially this is going to produce our cerebral spinal fluid, cerebrospinal fluid. So obviously very important to our bodies. Now, one side, again, always is going to be covered in the cilia. This is the side exposed because that's basically what's helping to move that fluid along. If we're literally producing fluid, we don't want to just leave it there. We want to produce it and we want to move it wherever it needs to go, including keeping our spinal cord and our brain vesicles lubricated. Next up, we have the microglial. So these are the tiny glials, right? You can see this guy right here, how small he is compared to the nucleus of an actual neuron, right? These microglials kind of look like, almost like the cell body and some dendrites of a neuron, but much, much smaller, much, much smaller. Now, these guys are really important for eating dead and damaged tissues. Hey, you're not supposed to be here. You're the garbage. I got to get you out of here. We need to keep things nice and clean. We need to keep this workspace tidy. That's what these guys are doing. They also can replicate, especially in response to infection, right? So if you do have that infection, they want to make sure that they are there. They are helping to clean things up, and therefore they are numerous in amount. Let's see, they are the least common in our central nervous system, so sometimes working harder than they should be. But they can travel throughout the central nervous system and go wherever they need to be to basically help do their job. Finally, we have the oligodendrocytes. These guys kind of are made of the same myelin sheaths in those Schwann cells that we just talked about, right? It's the same myelin sheaths. It's just wrapped. And just like the Schwann cells, they're used to insulate. They're used to speed up these signals. So we're basically helping to facilitate them. By wrapping this around, we're providing insulation, we're speeding up our signal, we're not losing any of that electrical signal elsewhere, we're basically directing it where it needs to go. And you can see it does have lots of little processes that come out And it's all, again, just that plasma membrane, that myelin sheath. It's basically just plasma wrapped. And that's what you can see right here, just wrapped in that myelin. We're not done. We do have a couple more. We've got satellite cells as well. So these satellite cells, as you can see right here, right, the little pink one, are basically going to be surrounding these little cell bodies, right? And they're going to help to provide support, nutrition. They're also going to separate each neuron from each other. So they kind of separate each of the cell bodies and they wrap around it almost like a little blanket. And they say, no, you go over here. You don't belong over there. You belong over here. And that's so we don't get our signals mixed up, right? You don't want any signals getting crossed there because then wonky things can start to happen. So that's what you do. You wrap around almost this little protective layer over here. And that's why they're satellite cells, because they're kind of just circling around these little cell bodies. Next up, you have some neurolemmocytes, which are also the Schwann cells. We did talk about these already, not quite a type of glial cells, but you can think of them like that, right? Especially because they look almost just like this guy, except it's this part right here. So, right, these little neurolemmocytes, also the Schwann cells, they do each wrap themselves around one axon only. So this is not connecting from axon to axon like some of the ones that we've seen so far. This is basically, hey, I'm only on this axon and I'm only wrapping around right here. And that's what you can see right here. And now remember, they do have that separation. Those nodes are Van Veneer, also known as those neurofibril nodes. Now, this again, helping to, we've talked about several times before, to provide insulation, right? And to speed up those signals. Very important that these signals fire as quick as they can. Otherwise, we're moving too slow, we react too slowly, and very bad things can happen. So let's now look at how we actually get these wrapped around our axon. Since they are just made of a membrane, right? We know that we can just fold as we need to. We can fold, we can fold, we can fold, we can wrap, we can wrap, we can do whatever we need to do to start, again, insulating and giving layer by layer by layer by layer. And that's what you can see right here. So you have the first wrap right here of the neurolemmocyte, right? That's the original creation of the myelin sheath. It's going to continue to wrap over itself, and then it's going to kind of start to roll around itself. Think about like a roll of wrapping paper, right? It's rolling and rolling and rolling and rolling. Instead of unrolling it, you're wrapping it up, you're rolling it up. And that's what we can see right here. So we start to, again, form multiple layers, these consecutive layers. As we keep overlapping, that multiple layers now becomes our myelin sheath, right? And we do still have our original neurolemmocyte nucleus because it's what we need for the cell. But this whole thing, technically, wrap, is still just one cell that's just wrapped around itself. And that, again, causes that insulation of these messages, which is so important to be able to send down our axon. Now, like I said earlier, because of these gaps, because of these nodes of Van Veneer, they basically are able to send signals faster. And you're thinking, like, how is that possible? If there's gaps in something, wouldn't it be harder to jump over? But in fact, no, it's known as saltatory conduction. And it's faster than regular conduction. Rather than just traveling in a straight line from one to the other, it basically goes, pew, pew, pew. Again, it's almost like, it almost like teleports from one to the other. It's jump, 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 jump. And so that's what it does. Instead of going it goes, and it like speeds up. It's almost like hyper speed, which is, just fascinating. All right, so we can't talk about our nervous system without talking about nerves themselves. So what is a nerve? In fact, when you hit your funny bone, you're not actually hitting the bone. Your bone doesn't have any feeling in it. It's just a bone. But what you are hitting is your nerve. And if you've ever seen the movie, was it 48 Hours? When the guy cuts his own arm off? Oh my God. I saw it, and when they get to that part, and it's almost, they do like a twang of the guitar, and it's like twang. It's like, that's what hurts the most. And it's because these nerves are innervated, guys. This is how you feel things. This is literally how you feel things. So they are cable-like organs that do run all the way through the length of your body. Very tough to get through. But they do have numerous axons wrapped around it. And it's, again, because this is our signal center. Essentially, this is our highway. So we do have these nerves with these myelinated axons, but we also have some that are non-myelinated as well. But usually the axon is always going to be surrounded by that Schwann cell. And again, it's because it's providing that insulation to make sure that we get our signal sent where it needs to go. So say we're that guy and he's cutting off his arm and that neuron's getting severed. Obviously not that intense, but say our neuron has been severed, right? So trauma has severed our axon and it's going to split it right in half. And now our signal is broken and we can't go anywhere and we can't send the signal to where it needs to be. So what happens? So essentially, right inside here, right, we have a breakage, a interruption of our signal that's being sent. So what's going to happen is this part is going to start to degenerate and it's going to start to disintegrate and it's going to start to break down because it's no longer useful. And so because this whole section can't receive any signals now, this is the important part of the cell. So we're going to start to break it down right here. And that's exactly what you can see right here is we seal it off and we end the axon. So we take this away and we say, hey, we're no longer needing this. We're going to break it off right here. We're going to stop it right here. Once we stop it, we were going to form what's known as a regeneration tube. And that's just that. It's a tube of regeneration. So it's going to start to grow and it's going to start to elongate again within the good, healthy part of that neuron, of that axon. And then it's going to start to reach back out over here to that skeletal muscle that had the damage. Once you finally get close enough, right, it's going to start remyelinating the axon. You're getting longer and longer and longer. You're getting more of those cells wrapping around, creating that myelin sheath. And then finally, you're going to get all the way to the end. And then you're going to finally be able to grab on to that skeletal muscle again and to, again, innervate to your little effective cell, which in this case would be your muscle tissue. Now, unfortunately, we can't always regenerate these parts. If you have damage enough that actually causes permanent damage to your nervous system, you can potentially be paralyzed. And that's what happened to actor Christopher Reeves, who famously played Superman and was Superman. When I was growing up, this was the Superman that we knew. And then through a horseback riding accident, he fell off his horse and he hit basically the back of his neck and he broke his back. And then he was paralyzed forever. So he was a huge advocate of neuroregeneration and research and donating money and giving talk and awareness about it. But he was paralyzed for the rest of his life. And it's sad because, like I said, these cells don't regenerate. And when they are damaged, they are damaged. And therefore, be careful, guys. Be careful with yourself. Be careful with your brains. Be careful with your bodies. You only get one. And it's very, very tragic when something happens, especially to someone who was like this, or really to anybody. But there's also normal disorders of the nervous system that can happen as well. Naturally, multiple sclerosis is one of those. It is an autoimmune disease. Essentially, what happens is it attacks the myelin sheaths around your axon. So your central nervous system is not able to have proper insulation. It's not able to send those signals where they need to go. And those myelin sheaths basically just start getting damaged and destroyed. So it does affect really wide, like it's super wide. You can have a more mild case, you can have a more of a severe case. It really depends on how much of your myelin sheath is destroyed. It does affect more women than men, which is one of the few. I feel like most of the diseases, not most, but a lot of them are kind of centered around men specifically. But when it is, and the least common when it is men, it is a little bit more disastrous and a little bit more devastating. It is more severe and quick. And we really don't understand why this happens, like why our myelin sheath gets destroyed. It could be numerous things, but we're still trying to figure this out. We're still trying to discover some of these things, and it is quite sad and very tough to deal with. So you not only have these physical processes, walking becomes harder, speaking becomes harder, but everything just becomes harder, and your body ends up working a lot harder because it's not getting the proper signaling that it needs to. It's not able to communicate to itself as well as it needs to, and it can lead to some pretty sad conditions. So again, take care of your bodies, guys. You only have one. Be good to it. Don't let this happen to you when they're crawling all over your myelin sheaths. Right? Anyway, guys, thank you so much. We have one more lecture to go. I'm so proud of all of you. Keep it up. We're so close. Thank you again, guys, and have a wonderful day. I'll see you guys in class.